Good afternoon, teacher. Today I'm going to be presenting on the lab number 4, the experiment. And in this section, I'm going to be covering the first experiment, which is on the hydrogen peroxide. Okay, so for this experiment, we want to control the rate of reaction of a foam reaction. So the reaction that we wanted to catalyze was the one of hydrogen peroxide. Hydrogen peroxide decomposes into water and oxygen. And we know that a catalase known as catalase is the one that helps decompose hydrogen peroxide into water and oxygen. And let's see, if it was not present, this decomposition would take so long, and this is due to the fact that the activation energy is very high, and so that's why it takes so long. So by adding catalase provides an alternate pathway with a lower activation energy required, and so the reaction happens at a very faster pace. So how can we control the rate of the reaction? By adding this catalase and by changing its concentration. And the catalase can be found in yeast, which is what we will be using today. We will be changing the concentration of catalyst to see how it affects the rate of reaction. And the rate of reaction, we will be measuring it by the amount of oxygen that is being released, which will be represented by the foam. So we will measure the foam with a ruler in the test tube, and that will be a proxy for the rate of reaction. Okay, so first, these are the materials that I used. I used 3 test tubes. I used pipe. No, I didn't use a pipette. I used this dropper that I bought. I used this soap, hydrogen peroxide at 3% concentration, a yeast packet, 5 cups, measuring spoons, ruler, timer, calculator, graph paper, pen, and towels. And also, in order to maintain the test tube standing up, I used flour. I just mixed some wheat flour with some water and kneaded, and it became this like very hard texture, and I used that to keep them standing. Okay, so the first thing that I did was prepare the yeast solution. So this I only had to mix the yeast, the 7 grams of yeast, with about half a cup of water, and mix it a little bit, and that's what it came out as this. Then I prepared the peroxide solution, the first peroxide solution, which is the one with the higher concentration. So in this part of the experiment, I put in a cup, I put half a teaspoon of soap, and then I added 2 tablespoons of the hydrogen peroxide. Then I mixed it without causing any foam. Then I prepared my test tubes. I put them, as I said, on the flour, on the wheat flour, standing up. Then I added 1 milliliter of the detergent peroxide solution to the 3 test tubes, which is this picture right here. I added it using this droplet, which measures exactly 1 milliliter at its fullest. So this is how it appeared after adding the peroxide solution. And then I added half a milliliter of the yeast solution, which is where we have the enzyme, the catalyst. So I added half a milliliter, which is half the capacity of that dropper that I'm using, to each one of them. And prior to that, I had timed my phone to 2 minutes so that I could start the timer as soon as I put the enzyme in the test tube. So this is how it started, and after 2 minutes, foam went up, way at the top. Just after 2 minutes, it went by very fast. So it went to the top, and then I had to measure this using a ruler, and these are the measurements. In test tube 1, I got 8.4 centimeters, 8.1 centimeters in test tube 2, and 8.0 centimeters, which altogether give an average of 8.2 centimeters. Now we change the concentration of the hydrogen peroxide. So instead of adding 2 tablespoons of hydrogen peroxide, we added The soap again, they have a teaspoon, and this time only 1 tablespoon of peroxide, hydrogen peroxide, and 1 tablespoon of water in order to change its concentration. And we follow the same steps. We added the yeast solution first, we added the peroxide solution first, then we had the timer prepared, and then we added the yeast solution, swirled them a little bit, and after 2 minutes, this was the foam. And as you notice, it didn't go up very high. And I will explain a little why this happened. I measured the heights, and I wrote them down here. So after that, I changed the yeast concentration. And the yeast, it was the end where the enzyme catalase is. So in this part, I did the exact same thing as I did before, just that this time I added more higher concentration of the yeast. So what happened is that I noticed that the foam was very, very, very fast, and it went to a higher, it went up very high. See, it almost overflowed the test tube to about 9 centimeters. So this was my result overall. In the second experiment with the lower concentration of the hydrogen peroxide, I got 5.5 centimeters in the first test tube, 5.6 in the second, 5.8 in the third, which give an average of 5.6. Then with the 1 milliliter yeast solution, these were the heights, which overall gave an average of 8.96, which I rounded off to 9 centimeters. So this is the graph that I drew, in which I plotted the hydrogen peroxide concentration versus the height of the foam, which in other words, which is the rate of the enzyme reaction in this case. So as you can see, they have a linear relationship. We can see that as we increase the hydrogen peroxide concentration, the foam height also increased at the same time. And you know, we would have seen it a lot more if we had done a lot more concentration, but since we only did 2, this is how our graph came out. So for my conclusions after observing everything that I've seen in this part of the experiment is that as we increase the hydrogen peroxide, which is our substrate in this case, the reaction rate increased. It increased. Say we would have done a more varying experiment with more different concentration, we would have gotten a graph like this, because after a certain time of increasing the substrate concentration, all of the enzymes would have been used up, and so it would have come to a plateau around here. It would have increased in a linear manner first, but then after there is no more enzyme available to catalyze the reaction or to interact with the substrate, then this plateau would have occurred. But since we only did 2, we got a very linear graph. And then the next thing that we noticed, by increasing the enzyme concentration, we also get a linear graph. This is because increasing the enzyme concentration allows for more interaction between the substrate and enzyme. So you're increasing the enzyme, so the reaction can go and go and go more and more. That's why it just keeps going up. So that's it for this part of the experiment, and those are my conclusions. Another question that I wanted to answer is, let's say that we wanted to find out, if we could find the hydrogen peroxide concentration for 3, 5, or 7 foam height. Perhaps we could use this graph and interpolate. Say, let's go from 3 centimeters to here, we would get around 0.245 hydrogen peroxide concentration if we wanted at 3 centimeter height. And for 5, we could have also done the same thing, gone from 5 here, we would have gotten about 0.38 moles per dm cube, or moles per liter. Now for 7, If we look at the graph, we would have gotten approximately 0.65 mole per liter. And that's it. Very important that we keep in mind that enzymes are very essential for our daily lives. Say, for example, if we didn't have them, many reactions would take so long to occur, which is why I think it's very important for regulating chemical reactions, especially in the body, because we need them, and without them, we would basically not be able to survive, or things wouldn't happen as the way they do right now. Thank you.